Hi, so today we're going to be going through another talk, and this is again continuing on with some of our lectures and sort of seminars regarding the concept of analyzing calorimetric data and calculating things like metabolic rate and correcting for covariates or nuisance variables. And I'm going to address one which I don't even think is particularly addressed well in the field as a whole, but we've now done some work on this. And this is a problem to do with weight loss in calorimeters, or as we're going to see first, weight gain. And it is a problem that can crop up in your experiments, particularly if your groups that you are working with have divergent body weight gains or losses in the calorimetry system. And this comes about because calorimetry can be a stressful environment. Now, modern systems are good at minimizing that. They use home cages. They have a lot of enrichment to try and reduce the stress on the mouse going through it, but it is not perfect. And you may find yourself in a situation where, say, your wild types have been weight stable and your knockouts have lost 1 or 2 grams of body weight on average across a calorimetry run. And you want to know whether your energy expenditure data is reliable and whether that negative energy balance is impacting on it. And I'm going to show you some data and a couple of examples of how we dealt with that, and also some other considerations you may have when designing your calorimetry experiments, depending on what you're trying to get. So one of the things that's the case now with modern calorimetry systems is you can try and get everything out of a calo run, but maybe it would actually be better to try and focus on getting what you actually need. And so what I would argue is that calorimetry is actually gas exchange. It's gas exchange in order to calculate 4 critical variables: VO2, VCO2, energy expenditure, and respiratory exchange ratio, or RQ. And conversely, I would argue not calorimetry would be variables which you can get out of systems from the likes of Sable, TSC, and Columbus, which are food intake, water intakes, body weights, and activities. And so why am I separating it like this? Well, the left side, you need a calorimeter. You cannot measure these kind of variables without some form of calorimetry system. The right side, you can do outside of a calorimeter, and you can measure them with non-calorimetric equipment. And it may be worth thinking about whether you want to get really good energy expenditure data, really good RER data, versus getting everything in one experiment. And in an idealized world, and when these systems work perfectly, you can do it. But when they don't, then that's when you run into a potential problem. And so the concept that you can get at this, everything from one experiment, has been popularized by the calorimetry manufacturers. But the problem is that in order to get all of these variables, compromises have to be made. You have to have devices in the cage to allow you to measure these things. And things like food intake, well, the food is not going to be in the normal home cage place. The water, it's going to have to be attached to a mass monitor. It's not going to be in the normal place. And whilst if you have really large amounts of calorimetry capacity, you can keep your animals in there long enough to acclimate them. Whenever you change an animal's environment, you're going to introduce some sort of stress or at least novelty. And so this means you're making a compromise when you're doing this. So then what does this manifest as? Well, the typical calorimetry runs that we've been doing in the IMS for perhaps the last 15 years, so I work in the Institute of Metabolic Sciences, it's 48 to 96 hours long, and they may include things like drug treatments and diet switches and all sorts of other interventions into the animals. Whereas the International Mouse Phenotyping Consortium took a very different approach. This was a transnational 10 centers system where they were trying to put every single knockout mouse line for every gene in the mouse genome through a series of pipelines to get 300 variables, and one of those variables was calorimetry. And they did it for 21 hours. So what you consider to be a very short period of time. And so the question is, which of these is better? And intuitively, you feel like more time equals more data equals more reliable. And this is certainly what I thought. But I'm going to actually argue that I think I've been doing it wrong for 15 years with these 48-hour calo runs. And I actually think the IMPC approach may actually be superior if you want to assess free living metabolic rate. And that's all you want to get from your animals. So I'll go on now to try and explain how I'm making this claim that I have done everything wrong for 15 years. And so what led to this epiphany that I am an idiot? And handily, my face is covering the word idiot. And it was a project I did with Tony Cole. And the project was on the NNAT mouse. So this was work with Tony Cole, Irene Chimino, and Debs Remington predominantly. And we were looking at a mouse which had a gene called NNAT knocked out, but it's kind of slightly different because this is a imprinted gene. So you actually only basically get inheritance, I think, down the maternal line. And so what can go wrong with long calo? And I think this is a beautiful example of how you need to be careful with longer calorimetry runs. And so one of the really amazing things about the NNAT lines, which is if you have the NNAT knocked out from the paternal allele, so you have then the animals, the offspring, will be biphasic for their body weights. So they're genetically identical. All of these are effectively NNAT knockouts because the maternal allele is wild type, but that's silenced. The paternal allele is gone, and you will get a bimodal distribution. Some of the mice will be somewhere between in the range of 30 grams, and they'll be relatively similar to the wild type animals. And then a proportion of them will be heavier, quite a lot heavier. I mean, in the original paper, and this was in the TRIM 28 haploinsufficiency paper here, which was in Cell from Andrew Pospasilic, we're looking at the peaks of these bimodal distributions, maybe about 15 grams heavier. So it's a big difference. Now, another group was working on these animals, and that is the group of Dominic Withers, who we've collaborated with from my lab, which was TVP lab. We weren't collaborating on this particular project, but what it showed was they looked at global knockouts. You've got, again, this NNAT negative P, meaning the paternal allele has been silenced, so they're effectively knocked out. And actually, they show something quite interesting, which is the body weights on average are the same as the wild types. But there's N to 7 to 14 animals per genotype. And actually, there's no good reason why it shouldn't be a 50-50 split. And this was because of this feature. What they showed was the big difference was not the average body weights, but it was the variance. And in their case, in their hands, what they found was they actually got a population of animals that were lean and a population of animals that were normal weight relative to the wild types. And the averages come out relatively similar, but in this case, you were seeing this bimodal distribution. And so the big difference is the variance. And the plus minus M are effectively wild type and show both similar body weight, but also similar variance to the wild types. And so we followed up on this as I say, working with Tony Cole and Irene to look at the neonating deficiency. And we found that this was what was driving this variability was a hypervariable food intake, which was driving the bimodal obesity. So that was the question. Was this difference in energy expenditure setting up these variable body weights, or was it differences in food intake? And we're going to try and argue it's food intake, and it's actually something even subtly different to that in that it's food intake driven by a response to a novel environment or a stress. Okay, so this was not a trivial question. And the reason it's not trivial is we can't genotype these mice. And it's a real pain. And so we have to come up with pretty crude ways of doing it. And so we had two populations based on two previous papers of NNAT plus minus mice, and in this case, the minus is minus P. I should have written minus P. And these were the lean group, which was running basically up to about 22 grams. I think these are the females. And then we had the obese ones, the other half, and they can run up to 45 grams. So, and what we effectively did was cut them in half and say, okay, this is where we see a sort of the first step change. And you can argue about whether these two should be in this group. It's very hard to do. And what we actually ended up doing was putting this data through things like PCA to say as many variables as possible. Where do they split out as groups? We tried k-means clustering, and we also ended split at 50% body weight, and they all gave the same result on females. A little bit different on males, but on females, everyone said this is where to cut your line. somewhere between mouse 11 and mouse 12. And these are the body weights of individual mice, so half and half, 11 and 11. And so what you get with that is the average body weights. You have the wild types. The lean NNAT mice are basically about the same weight, and then the obese NNATs are really substantively heavier, something like 8 grams heavier than the lean ones, about 7 to 8 grams heavier than the wild types. So quite a big difference in body weight. But something remarkable happens when we put them in the calorimeter, and we'll go back to this, is the change in body weight of these animals is staggeringly different. So the wild types are basically weight stable. Our lean NNATs pretty variable, and again, we don't really know exactly where we're cutting it, but they gained about a gram in 48 hours. The obese ones, they gained nearly 4 grams on average in the calorimeter, which is pretty spectacular. And unsurprisingly, we see a huge difference in food intake. And so obviously we've got these big weight gains and we've got these big changes in food intake. What we wanted to know is there any component of energy expenditure in the equation here? But you get a problem, because you now have to factor in 2 covariates into your system. First of all, these mice are heavier, and as we know from our previous talks, larger mice expend more energy. And so we have an energy and expenditure versus body weight plot, and we can see the obese NNATs are looking hypermetabolic-ish. These gray dots in this line is looking like it's above the blues and the oranges. The blues might be a little bit above the oranges, and those are the lean NNATs. And then we can see here again, the lean NNATs in blue, the obese NNATs in gray, and the wild types in orange. And we're plotting, in this case, change in body weight, which range from -3 grams in some of the wild types up to +6 grams in 48 hours for some of the obese NNAT animals. And again, we can see the energy expenditure for the delta body weight is offset here, but it's partly offset because these animals are bigger. So we're going to need to put both of these into our model simultaneously so we can simultaneously account for these 2 covariates and make sure that once we've corrected for one, we don't lose the effect of the other. Because these are obviously larger mice, which are also gaining more weight. So it could be the fact that everything reduces down to really, it's just the biggest mice gain the most weight. And if we do a traditional ANCOVA for metabolic weight using just body weight as the covariate and ignoring this change in delta body weight, we see something which I think is not necessarily unexpected. We have estimated marginal means, body weight as the covariate, and we see that the wild types and the lean NNATs are largely the same, but we have a very significant effect with the NNAT obese. And just note here, the p-value for body weight is 0.044. Okay, so it's significant, but only borderline significant with the NNAT obese animals having really a much higher metabolic rate according to this. But there is a phenomenon in all organisms, which is if you eat in excess, you eat above your requirements for body weight maintenance, you will increase your energy expenditure. And this will be through 2 potential mechanisms, which is SDA, which you can think of as thermic effect of feeding. So the fact is to process all this food, for the guts to squidge it through, and then for the biochemical transformations, because if these animals are on chow and they're getting 4 grams, that's not glycogen. They're turning that into fat, and that is going to be producing heat as a byproduct. And heat is what we're measuring with the calorimeter. There may also be this phenomenon known as Lux's consumption, which is more popularly now known as dietary thermogenesis, which is a burning of excess energy in brown adipose tissue. It's debatable, but how much of these components are impacting. But the concept is increasing energy expenditure above what is required for homeostasis will drive an increase in energy expenditure. So the question then was, could we account for this effect of the change in the body weight as well? And what we could do then is we could put in a second covariate, delta body weight, the change in body weight, to capture this weight gain. And this was highly, highly significant, as you've seen from the graph. But the critical point here is the body weight significance improves substantively. Body weight becomes a better predictor. It explains more of the variance when you also know the delta body weight is in there. And this tells us that body weight is not purely correlated with change in body weight. It's not just the biggest mice that gain the most weight. What you need both covariates, and when you have both of them in the model, your model gets stronger, and it gets substantively stronger. I think the adjusted R squared for the entire model here is 0.858, and I'll flick back from 0.762. So adding this covariate, we're basically explaining another 10% of the variance, and that's pretty big. That's pretty substantive. So that's pretty cool. And now what we see is our genotype. I mean, they're not strictly genotypes, but group effect will be more honest, because this is actually having 3 different variables, not comparing just wild type to all of these together. It goes away. There's a tendency to higher metabolic rate in the NNAT obeses, but it's nowhere near significant. And the difference is down to sub 2 joules, and that's well below, that's below 7%. So there might be something very subtle there, but this enormous effect we were seeing on energy expenditure, or metabolic rate, more accurately, calculated using just body weight, the traditional method that we're always told to do by things like CHOP 2011 paper and by my own talks, which was showing actually a boost of well over, in this case, well over 5 joules per minute per mouse on a starting value of about 26. That's gone. That's gone. So this 20% boost in energy expenditure on metabolic rate is gone. Okay, and we can look at how this pans out, because these are the mouse, the wild type, least weight gain, NNATs, bit of weight gain, the lean ones, obese ones, loads of weight gain. So with body weight only, we get this big effect, which you can see with the red arrow here, but including both covariates, we've largely eliminated this big change in metabolic rate. And why is this important? I mean, obviously, this change in body weight is a real thing, okay? And it's driving some effects on energy expenditure. But we were certain this was not happening outside the calorimeter. And the reason we were certain, and I'm going to actually move my picture so you can see this, is that we had set up a 9-gram difference between the wild types and the obese NNATs in 4 months. The difference between the wild types and the obese NNATs in the calorimeter was 2 grams per day, or 60 grams a month, or 240 grams, which is an impossible weight for a mouse, over the course of those 4 months. So what was happening in our calorimeter was not representative of what was happening in free living life. And so to get an energy expenditure version, we wanted to correct out this huge difference in body weight gain that was happening over our calorimetry run. And so we could come down and really start to think that it's food intake. Interestingly, the fact that when you put the animals in a calorimeter, which is a slightly stressful environment, wild types go into very subtle negative energy balance, they gain all this weight. Well, actually, we had another test, which was like an open field test, and that was really demonstrating that, yes, this is a response they would do to a stressful or novel environment. And it may be much of this weight differential that's being set up is through things like cage changes. Rather than this being homeostatic control of body weight, this is actually something really quite cool, which is like, how do you respond to stress? Well, there are people who respond to stress by eating chocolate bars, and they gain weight in stressed environment. And we think this potentially could be a model whereby this genetic mutation or variants in this gene in humans could be something that predisposes you towards obesity driven by things such as stress or changes in environment. And so we ended up coming up with this conclusion. NNAT deficient mice maintained the hypervariable body weight phenotype with the calorimetry system. Food intake in the NNAT plus minus P mice was hypervariable, with some mice consuming more than twice the intake seen in wild type mice. Littermates. And a hyperphagic response was also seen with the NNAT plus minus P mice in second non-home cage environment. That was the test I was telling you about. And the expected correlation between body weight and energy expenditure was seen, but correction for the effects of positive energy balance and body weight greatly diminished the effect of the neuronitin deficiency on energy expenditure, and in fact rendered it non-significant. Okay, so that worked for positive energy balance, but what about negative energy balance, which I think is a more common problem? And so do mice which lose more weight than others lower their metabolic rate proportionally? Seems reasonable. So this experiment is an interesting one. It's kind of a fortunate event that happened. And it was 2 calorie runs, 48 hours each, and the mice were on 60% high-fat diet. And the 2 runs showed really very different energy balances. So we got weight loss in the first run, and that was of about 0.5 grams. And in the second run, the average weight loss was about 0.28 grams. So really big difference in weight loss. And what was different between these 2 runs? And this, by the way, was half wild types and knockouts in the first run, half wild types and knockouts in the second. And the run with the lower weight loss had food and gel patches in the cage for 7 days prior to the calo to acclimate the mice to new locations of food and water. And that was kept in there whilst they were going through the calo. But this raises an obvious question. This is insanity. Why would you change conditions halfway through a study? This is like the last thing you should do in science, is take half your animals and change all the conditions before you do your second batch of animals. Well, the answer to that is the following. In the United Kingdom, we have very strict animal legislation, and there are limits on how much weight loss we are allowed to have. And you do not want to get a letter from ASRU, Animal Scientific Regulation Unit, enforcement. And these people don't muck about. It comes on headed letter paper from the Home Office, and they consider exceeding a weight loss of 10% on the protocol. This is not in relation to the mice that I've shown you. This was my own, is not acceptable. So you need to bear in mind that there are considerations beyond just the science you're doing. Not getting prosecuted is important. Okay, so let's have a look at the data from these runs, and let's plot a body weight energy expenditure graph. And in here, you've got the orange are the low losers, the mice which lost less weight, and the blue is the high losers, and those are the mice that lost more weight. And you can see there is actually an offset between the 2 runs, and the higher losers have a lower metabolic rate. Body weight, energy expenditure, this line is below. And actually, what you can see is the energy expenditure using only body weight as the covariate in terms of the estimated marginal means is about 5% lower in the high losers than it is in the low losers. It's not massive, but it's going from maybe 0.6 on average in the low losers to about 0.63 in the high losers. And this is a small difference, but what it will definitely do is ram a load of noise into your analysis. So if your wild types may have a slightly higher energy expenditure than your knockouts, if you then pile a load of noise on top of that, it's going to be difficult to determine whether there's actually a difference of genotype. Now, if we do a Ancover analysis using body weight and delta body weight as covariates, estimated marginal means come into a point where there is no actual statistical difference between the runs. So it eliminates the statistical difference between the runs. And now they're all pretty much the same, maybe at most 0.1 joules between the 2 runs, if that, sorry, 0.01 kilocalories per hour. We're in kilocalories per hour country here. And what you can see is that when we look at the Ancover in terms of kilocalories per hour, there is no genotype effect. Average body weight is highly significant. It's run onto another line, 6.436 times 10 to the minus 5. And delta body weight is significant, 0.01. So it's a valid Ancover analysis. Okay, so this is all very statistical. And one of the problems with it is unless you have large numbers of animals, you only need 1 or 2 mice at most to break these delta body weight relationships. And you only need 1 or 2 mice to break the body weight relationships. And now you've got 2 different covariates and they can both get broken. And that's a problem. So is there another way? Okay, so what we looked at was the effects on energy expenditure due to negative energy balance increased over time. So you can see the orange lines here and the blue and orange lines are largely similar, pretty much up until about here. There's a bit of divergence in this period, and then they really start to diverge as we go later into the runs. And you can see the whole of the second day phase and the whole second night phase. That's where the big point of divergence is, with a little bit in the first night phase as well. And actually, if we split this run into the first 21 hours and then the rest of the run, what you can see is that the differential in terms of the 21 hours versus the rest of the run, for the low losers, they're fairly similar. There's a 0.04, but that differential in terms of how much energy the mouse expended between the first 21 hours and the rest of the runs, when we look at the high losers, that actually increments to 0.07 joules. So in other words, the longer you are running the calo, the more the negative energy balance is coming into play. And so then the question is, how robust is 21-hour calo data for energy expenditure? So the data from the IMPC. And here we're going to use Wellcome Trust Sanger Institute data from the mouse genetics program. And this is the MGP pipeline. So these are mice on a Western diet. And the first thing to know is over these 21 hours that they've had these mice in the calo, we can look at the change in body weight, and you can see that there is a huge amount of weight loss. There were 2 mice that did not lose weight. In fact, they gained weight. There was 1 mouse that didn't lose weight, and every other mouse, 190 animals, lose weight up to 2.5 grams. And you can see the energy expenditure along the y-axis in joules per minute per mouse. In this case, we're back to joules per minute per mouse. Sorry about the units. And you can see empirically, there is no relationship between change in body weight of these animals and energy expenditure. Well, okay, that's possible, but it may be the case that there is a subtle effect because we haven't included body weight, which is highly significant as a covariate in this. So we can now run the statistic analysis, and we can say, okay, we've included body weight in our analysis along with delta body weight. And then we can look and see, and this is a linear regression model because we have no categorical variables. We've included body weight in the system, and body weight has a p-value of 2 × 10⁻31. So it's hugely significant as a predictor of energy expenditure. But even with 193 mice, we do not have effects of delta body weight on energy expenditure. So it is empirically not predicting the energy expenditure of the mice once the mice have been corrected for body weight as well. So essentially the metabolic rate is not being affected over 21 hours by this weight loss. So this is okay because we've got a really big spread. So to try and really break this and really basically find a difference for the purpose of breaking the system, what we did is, okay, well, that's fine. But what if you have a situation where you've got extremes of weight loss versus weight gain, or at least weight stability to an extent? So we took the 20 largest losers, and we took the 20 smallest losers, including 2 mice that gained weight and 1 that was weight stable. And we asked, for these animals, okay, if we take these as groups, can we find a difference between these groups? Is there anything different between these animals? Okay, so weight gain. Obviously, weight gain is, by definition, on average, going to be very different between our 20 biggest losers and our 20 smallest losers. And in fact, it ranges from -2 grams over 21 hours. So that's a big weight loss. That's 2 grams a day. So that's equivalent to the NNAT split in negative energy balance. And for the smaller losers, it was about 0.4 grams. So not perfect, but getting close to weight stability. There was no difference between these animals in starting body weight, which is important. It wasn't the case big mice lost more weight, small mice lost less weight. And there was no difference in energy expenditure corrected for ANCOVA. So this is metabolic rate, energy expenditure corrected for ANCOVA for these mice, nor raw energy expenditure. And that makes perfect sense because you can see the starting body weights are identical, metabolic rates are identical, energy expenditures are identical. RER obviously is massively different because these mice that are losing weight while the energy expenditure is the same, they're eating less food. And if they're eating less food, that means they're burning their own fat reserves. And these RERs of these mice were getting down to close to 0.7, about 0.73. And this is probably pretty close to the minimum you can achieve in terms of respiratory exchange ratio in mice. Conversely, the ones which were losing far less weight were at about 0.78. So they were not as low. And this follows that there was food intake that was going to be very different. But in other words, empirically, no impact on metabolic rate over this short-term run. So what are we going to conclude from this? So studying mice in a calorimeter for shorter periods of time can remove the effects of negative energy balance on metabolic rate between your groups. And pre-existing longer data, 48-hour plus calo data, may be correctable for negative or positive energy balance using regression-based techniques. And I stress this may be correctable because you're using two covariates. You need them both to be significant if you're going to be able to do these corrections. And that's not always going to happen. You're going to need good N numbers. And like with the NNAT project, it was nice because we had large numbers of animals, given that we were looking into this bimodal thing. You needed more than your N of 8 and 8. But it may be possible, and it's certainly worth trying if you're finding that what you're seeing in your calorimetry in terms of weight gain or weight loss is in no way mirroring what's happening outside of it. And, you know, it's pretty obvious. If your mice are gaining 2 grams in 24 hours, that's not happening every single day of their lives. Because, you know, they're not going to weigh 60 grams more at the end of the month. Not even an OBOB is going to pull that trick. And so this is a tool that may be of use in certain circumstances. And those circumstances, again, are where you have radical divergences in your groups in terms of their response to weight gain or weight loss. And I would argue, finally, you should make a choice as to what your primary outcome variable is. And if you want metabolic rate, then calo is an essential tool. And your experiment should really focus on getting the best possible calo data you can. You should be prepared to sacrifice things like activity measurements if you really need the energy expenditure by putting in environmental enrichment to reduce the stress. The food intake and water intake, we saw where we had food on the cage floors and gel pouches. You'll lose your food and water intake from the hoppers because the stuff on the floor of the cages, the mice can go eat. But you will reduce the weight loss and you will reduce the negative energy balance, and you may get longer term, more representative energy expenditure of the free living environment. I mean, the ideal world is if you can keep your mice in the calorimeter for days or weeks and they become fully acclimated to it and it just becomes their home. But many facilities don't have the luxury of running super long-term calorimetry experiments because the equipment is in demand. And you may also want to think then, if you're being forced to do short-term runs, then also measuring other things like food intake, behavior, etc., using other approaches to complement getting this energy expenditure to metabolic rate data, where having a relatively short period of time may actually be more suitable. Okay, so what I would argue then, finally, is calorimetry. Just remember, calorimetry is your gas exchange, because that needs a calorimeter. And the other variables you get from the commercial systems, they are not calorimetry. They are nice to have, but if your absolute critical variable is that energy expenditure, then, or that respiratory exchange ratio, then think about your design of your experiment to optimize what you are doing. And finally, I'd just like to acknowledge a few people. I don't normally do these, but obviously a lot of this work was principally from Tony Cole, Debs Remington, and Irene Cimino, provided all the NNAT information. And then also to thank Chris Elliott, who, when he was working at the Wellcome Trust Sanger Institute, provided us with all the raw data from the MGP pipeline that we were able to do those analyses, splitting the animals up. And the DMC team, who run all of the experiments and have run all the calo, which the data is what we're analyzing. So thank you very much.